Hello my dear students, this is your host Dr. Syed. Today let us study about the mode of reproduction in microorganisms. So here I have grouped the mode of reproduction of bacteria and fungi. The bacteria reproduces by binary fission, conidia formation, budding, cyst formation, endospore formation, conjugation, transformation, transduction, etc. And in fungi, you can see fragmentation, fission, budding, and fungi also reproduces via asexual spore, anthrospore, blastospores, chlamydospores, conidiospores, and via sexual reproduction, plasmogamy, karyogamy, and meiosis. You might be wondering why these pictorial diagrams are here in this slide. In order to remember the mode of reproduction in bacteria, you need to remember this simple sentence. In bus, you need to give cash with exact change to buy ticket from conductor. So the B stands for binary fission, C stands for conidia, exact stands for endospore, change stands for cyst, B stands for budding, T stands for transformation, C stands for conjugation. So most of the reproduction process by bacteria is covered in this sentence. When you come back to fungi, Facebook, F stands for fragmentation and fission, B stands for budding, and A, B, C blocks: asexual spore, anthrospore, blastospores, chlamydospores, conidiospores, and sexual reproduction by plasmogamy, karyogamy, and meiosis. You need to remember the movie PK. P for plasmogamy, K for karyogamy, M for meiosis. So as we discussed, the bacteria reproduces both sexually as well as asexually, and here are the 5 types: binary fission, conidia formation, budding, cyst, endospore formation, sexual reproduction, conjugation, transformation, and transduction. Moving on to the binary fission, here you can see in this slide, during the process of binary fission, the cell elongates itself and there is a replication of genetic material present inside the bacterial cell. Once the genetic material is replicated, there is a formation of wall, and this wall grows deeper, the cytoplasm is divided, and there is this growth of septae or a wall continues, and there is a formation of 2 daughter nuclei or 2 daughter copies of the cell, and this divides, detaches, and 2 new cells are formed. This process is called as binary fission. Going on to the conidia formation, here this is especially seen in the case of actinomycetes. We will take streptomyces as an example, which is a member of actinomycetes, and the actinomycetes are having the characteristics of both bacteria and fungi, which are capable of growing by producing mycelium. So here you can see a hyphae or mycelium, and at the tip you can see the conidia, and this is called as conidiophore, and this is called as conidia, and this conidia detaches from this and try to germinate when it encounters suitable substrate. Next comes budding. During the budding process, here you can see the cell, the bacterial cell. At one end there is a formation of bulging, and this bulging results in the formation of a bud. The nuclei, part of the nuclei enters to this bud along with the cytoplasm, and here the bud try to develop a wall and becomes an independent cell, and this bud try to multiply even though it is attached to the mother cell. Here you can see, and there is a formation of different cells, and at some point of time the partition wall gets detached and each individual cell starts germinating. Cyst. This is a special condition. This happens only when there is an unfavorable condition, and during the process of cyst, if you can see here, there are different cells, and when there is a lack of nutrient, when there is unfavorable condition for multiplication, this, in order to survive, forms a thick wall around its cell, and this thick wall makes them more dominant. And during the favorable condition, this entire cell, which is called as cyst, starts multiplying. This can be observed in the members of Azotobacter, which are reported to be nitrogen fixer. Next, formation of endospore. The name itself indicates endo, inside the cell, endospore. Here you can see a vegetative cell wherein there is a formation of, there is a partition, and this partitions try to grow by developing a spore coat, and this spore coat gets thicker and thicker, and there is a desiccation of the cell. The entire genetic material is within the spore coat, and this cell wall of the bacterial cell gets disintegrated, and the freely present endospore, when comes in contact with substrate under the favorable condition, starts germinating. So cyst and endospores happens only during the special cases. Next comes the sexual reproduction. Here the bacterial conjugation is a process wherein two bacterial cell comes in close proximity, and there is a development of conjugation pilus, and this pilus is formed between two cells, and the plasmids and other genetic material is transferred from one cell to the another cell. The cell which is transferring the genetic material or the plasmid is designated as F plus cell or donor cell, and the cell which receives the plasmids or other genetic material is called as recipient cell or F minus cell. Next comes the process of transformation. Here, during the process of transformation, the bacteria absorbs the genetic material or DNA fragments which are released by the other bacteria. Here you can see an example. The bacterial cells, here there is a bacterial cell and the cell wall is ruptured. The DNA present inside the bacterial cell gets fragmented, and each fragments are released to the surrounding media. The neighboring cell present in the media try to absorb this part of this genetic material which is integrated into its genetic material. Next comes transduction. The transduction is a process wherein it happens with the help of bacteriophage. The bacteriophage try to infect the bacterial cell, and with the help of the host machinery and the genetic information of host, it tries to multiply. It captures some of part of the genetic material of the host and try to multiply, and at the sufficient period when there is optimal number of viral component, they try to assemble inside the host cell and gets released. And when they are released, the newly integrated genetic information obtained from the bacteria is integrated into these newly released bacteriophages. When they infect the other cells, there is a transfer of genetic information which they had carried from this cell. So next, let us move on to reproduction in fungi. In fungi, the reproduction takes place via three modes. One is vegetative reproduction, second one is via asexual spores reproduction, and a sexual mode of reproduction. And in vegetative mode of reproduction, one of the most common method is fragmentation. The process of fragmentation, the hyphae breaks up into a smaller fragments accidentally or otherwise. They they break up the mycelia into smaller fragment, and the individual fragment try to develop into a new mycelium, new mycelia and fungal cell. Moving on to the asexual reproduction in fungi, it happens via fission of somatic cell, budding of somatic cell, and via asexual spore formation. Fission. In binary fission, a mature cell elongates and its nucleus divides into two daughter cells, which we have also seen in the bacteria, the mode of reproduction, and there is a division of genetic material, and there is separation of cytoplasm, and each the elongated cell results in the formation of a wall in the middle, and this wall separates the entire nucleus and the cytoplasm to form two identical daughter cells. And budding is a process wherein a cell wall gets bulged out and the genetic material and cytoplasm, or along with the cellular content, moves to this bud part, and this bud gets mature, and there is a separation of bud with the help of a wall, a septate, and this bud tries to detach from the mother cell and grows as an individual cell. The best example is yeast. Asexual spore reproduction in fungi. Different fungi produces different types of spores. Here you can see in this picture, you can see a rhizoid and the sporangiophore. At the tip of the sporangiophore, you can see sporangium. This sporangium consists of sporangiospore, and this sporangium gets ruptured and the sporangiospores are released, and individual spore try to germinate into a new specialized hyphae. The example is rhizopus. Next comes conidiospore. Here there is absence of sporangiophore. Instead there is the conidiophore, and at the tip of conidiophore you can see medulla, and this specialized structure medulla consists of phyallid, and this phyallid consists this phyallids. At the tip of the phyallids you can see the chain of conidia, and this conidia is nothing but a specialized spore, and this conidia, when they gets detached from the fungi, try to germinate into a new fungi. The example for this, Aspergillus niger and Aspergillus fumigatus. Anthrospore. And in anthrospore, it is a very primitive type of spores formed by breaking up of fungal mycelium. Here you can see a fungal mycelium which gets breaked into smaller fragments, and this smaller fragments develops into a new hyphae. The example here is Geotrichum trichosporum. Chlamydospore. Here, this is a special condition wherein within the mycelium, during the unfavorable condition, there is a formation of a spore, and this spores results due to lack of water content, and once this spore is developed, the wall gets thick, and once they acquire a favorable condition, this spore try to germinate into a new fungi. Example, Candida albicans, different members of ascomycetes, basidiomycetes, etc. Blastospores. It is a budding spore usually formed at the terminal end of the hyphae. Here you can see there is a formation of chlamydospores, and along with that chlamydospores you can see a bunch of spores in the form of blastospores, and these blastospores try to form branches, and even this happens only under the unfavorable condition. This is observed in the members of ascomycetes, basidiomycetes, zygomycetes. The sexual reproduction in fungi. The sexual reproduction is carried out by fission of compatible nuclei from two parents at a definite state in the life cycle of fungi. The process of sexual reproduction involves three major phases. The first phase is plasmogamy, fission of protoplasm. Second phase is karyogamy, fission of nucleus. The third phase is meiosis, reductional nuclear division. Here you can see in this picture the life cycle of fungi, both asexual and sexual reproduction. You can see the mycelia, which undergoes mitosis, which results in the formation of spore, and this spore try to germinate individually, and this entire process is called as sexual reproduction. Whereas this mycelium undergoes plasmogamy, wherein haploid cells from two different mycelia fuse to form heterokaryotic cell with two or more nuclei. This heterokaryotic stage undergoes karyogamy, the fusion of nuclei to form a diploid zygote, and this zygote undergoes meiosis to form spores, and this individual spore try to germinate. So with this, let's conclude this session. And my dear student, the mode of reproduction in fungi and bacteria are the important questions. I wish you good luck for your exams. Thank you. See you in the next video.